Hello, good afternoon everyone. So I am back with another session of Power Hour. I am your zoology mentor and guide for today's session again, and I will be taking up the chapters which is human reproduction, reproductive health and human health and disease. So like every session that we have had in the past, we will be discussing the important topics which are going to come for the NEET exam. Okay, so let us see what are the ones that we are going to take. Now these are the topics from where there are large number of questions that are asked every year, and there are few tricky questions, like we will be talking about human reproduction chapter where we are going to talk about gametogenesis. So we will see one numerical also. Yes, very well heard. It is the numerical that we will be talking about in this chapter with respect to spermatogenesis and oogenesis. We will be talking about menstrual cycle. This is the part where some of the students find it a little tricky with respect to the hormonal control. So that is also what we are going to discuss. We will then move on. We'll then move on to reproductive health, where we'll be talking about birth control measures, and then finally with health and disease chapter where we'll be talking about immunity, and we'll be having a rough idea about the drugs, because again drugs is the part where they can be matched the columns that are coming and that can come in our exams where you might tend to make a mistake. So let us see how not to make those mistakes and how to correct and get good marks. Okay. So now when we are talking about human reproduction chapter, as you are well aware, human reproductive system is basically, since we are unisexual animals, there is male reproductive system and there is female reproductive system in separate bodies. When we are talking about male reproductive system, I'll just give you an overview of the entire reproductive system before we move on to gametogenesis. So this is the diagram 2.1a of our NCERT page 27, where they have given a diagrammatic sectional view of the male reproductive system, where you can see the primary reproductive organ, that is testes, which is situated into a sac-like structure called scrotum, which is then followed by the accessory reproductive ducts. You can see I'm highlighting them, followed by the accessory reproductive glands. So you can see there are seminiferous tubules here, there is prostate gland, there is Cowper's gland which is present here, and there is a part which is a part common to urinary system as well as reproductive system, which is the urethra, passing through the external genitalia, that is this part, which is called as the penis. Now since urethra is a common passage for reproductive as well as urinary system in males, we call male reproductive system also as male urogenital system. Okay. Now as we said that the primary reproductive organs are testes. What is the main function of testes? We can see the same diagram here also. This is another view, which is the ventral view of the male reproductive system, where again we can see that this is the primary reproductive organ, that is testes. Now testes which are there, as I told you, the entire male reproductive system is situated in the pelvic region, except testes which are situated outside the abdomen into a sac-like structure which is called as scrotum. Now why are testes situated outside the abdominal organ in the scrotum is because testes need a temperature which is 2 to 2.5 degrees Celsius below or above. What do you think? You've already studied, so quickly tell me, it is below or above the normal body temperature? Yeah, good afternoon Shrutika. Yeah. Is it below or is it above the normal body temperature? Yes, very good. It is below the normal body temperature. So 2 to 2.5 degrees Celsius below the normal body temperature, because that is the temperature essential for the purpose of the spermatogenesis, one of the important functions of testes. There are two functions that testes perform. One, which is production of the male gametes, which is called sperms, by the process called spermatogenesis, which we are going to discuss in detail. And second is produce male hormones, which are sex hormones, which are called as androgens, and the main one of them is testosterone. Now remember, till now you were studying in school that testosterone and androgens are synonymous with each other. But even if you go back to your chemical coordination chapter, it is mentioned beautifully that male male reproductive system, that is testes, produce a group of hormones which are called androgens, and the main androgen among them is testosterone. So here we are not going to focus on the functions of testosterone or androgens. We are going to focus about your spermatogenesis process. Now, where does spermatogenesis take place? It takes place in the testes. Now, if we are taking a sectional view, we know that within the testes, there are small compartments, and these compartments are called as testicular lobules. In each lobule, there are 1, 2, 3 highly coiled tubules, which are called seminiferous tubules. These seminiferous tubules on the inner side, remember this can come as an MCQ, are lined by cells which are called as spermatogonia or male germ cells. It is these cells which will undergo mitosis and meiosis eventually and form haploid cells which are sperms. Okay, how do they form it? Let us see. Now what happens when we are talking about puberty? At puberty, from the hypothalamus, there is a hormone called GnRH that is released, that is gonadotropin releasing hormone. Now this GnRH, which is released from the hypothalamus, goes to the anterior pituitary. Where does it go? It goes to the anterior pituitary, and in the anterior lobe of pituitary, it stimulates the cells to release hormones which are FSH, that is follicle stimulating hormone. We've studied this in the last lecture, if you recall the last lecture, okay? And it releases another hormone called LH or luteinizing hormone. Now this luteinizing hormone which is there goes to the cells which are called as Leydig cells present in the testes. These Leydig cells, if you see in this diagram, this is a seminiferous tubule. Okay, this is another seminiferous tubule. Now in between the seminiferous tubule, the space that you can see here, this is interstitial space. In the interstitial space, there are clusters of cells which are present, and these are called as Leydig cells or interstitial cells. LH from the pituitary will come to these Leydig cells or interstitial cells, and in the Leydig cells or the interstitial cells, they will bring about synthesis and secretion of hormone which is your androgen. Now this androgen will go to the seminiferous tubule where there are these cells which are spermatogonia, stimulate the spermatogonia to go into cell division. How do these undergo cell division? Now once they go to spermatogonia, they will stimulate the spermatogonia to undergo the division which is mitosis. Now, spermatogonia will start into mitosis. Some of the spermatogonia, not all. Please remember, some of these cells is what is mentioned as an NCERT statement. Some of these cells which have undergone mitosis now will undergo differentiation to form what is called as the primary spermatocyte. Now, if we talk about the chromosomal number simultaneously, my dear children, tell me what will be the chromosomal number of spermatogonia? Yes, Gurkiraj, Shrutika, Mrunal, Harshali, Rupesh. Yeah, can you tell me what will be the chromosomal number of spermatogonia? Yes, it will be diploid cells. Very good, my dear children. So these cells are diploid cells. Now in cell division in botany, you've studied then when mitosis takes place, mitosis is also called as Equational division. So it will produce cells which will have equal or same number of chromosomes as the mother cell which is undergoing cell division. So spermatogonia is diploid. My dear children, if spermatogonia is undergoing mitosis followed by differentiation, then the primary spermatocyte will also be diploid cells. Yes. Now these primary spermatocytes will now undergo first meiotic division. Now first meiotic division, if you remember, is reductional division. Yes. Link it with your cell division chapter. Now when it undergoes the first meiotic division, which is reductional division, the primary spermatocyte, which was a diploid cell, say for example, we have one primary spermatocyte. This one primary spermatocyte, which is diploid cell, will now form secondary spermatocytes. How many secondary spermatocytes will be formed? There will be 2 secondary spermatocytes will be formed, and these secondary spermatocytes are now haploid cells. So these will be n/2. These secondary spermatocytes will now undergo second meiotic division to form what is called as spermatids. Second meiotic division, tell me quickly, is equational or reductional again? It is equational or reductional division? Yes, it is equational division. So again, you have n into 4. Yeah, so we will have 4 spermatids which will be produced. Now you see, these are 4 spermatids which are produced. But do the sperms look the same as the spermatids? No, they have a different structure. So the chromosomal number is attained in the spermatids, but these now have to undergo further process of differentiation, converting spermatids to spermatozoa. And this process of conversion of spermatid to spermatozoa is the process which is called as spermiogenesis. What is it called? Spermiogenesis. Are you understanding? And who helps in this spermiogenesis? Now see here, dear children, we had written that GnRH acts on the anterior lobe of pituitary, and that also brings about release of FSH. Now this follicle stimulating hormone is going to act on Sertoli cells and bring about release of certain factors, and these are the factors which help in the process of spermiogenesis. Are you understanding? Yes, is it clear? So we have one primary spermatocyte which is diploid, which undergoes the first meiotic division to form the first haploid cell category, which is secondary spermatocyte. So one primary diploid primary spermatocyte forms 2 haploid secondary spermatocyte, which undergoes second meiotic division to form 4 haploid spermatids, which undergoes spermiogenesis and forms finally what is haploid 4 spermatozoa or sperms. Are you understanding? So we have one primary spermatocyte. Now pay careful attention to this and remember this, will form 2 secondary spermatocytes, will eventually by division form 4 spermatids, and these will then form 4 sperms. Is this absolutely crystal clear? Yeah? Okay. Once sperms are formed, they will remain attached to the specialized cells called Sertoli cells. These cells that you can see here, with their heads embedded into the Sertoli cells till the time there is a signal for them to be released. And this process of release of the sperms is called as spermiation. Are you understanding? Now this can come as your MCQ. Spermatogenesis is formation of haploid male gametes called sperms or spermatozoa. Spermiogenesis is conversion of spermatids to sperms, and spermiation is release of sperms. Now keep this in mind because this can come as your MCQ or true and false statement or question where there can be fill in the blanks, and you can have that question where you have to choose A, B and C in appropriate fill in the blanks. Is that clear? Yeah. Now there's one more important part about this is where the human male ejaculates approximately 200 to 300 million sperms per ejaculation. Now there's only one sperm that is required for the process of fertilization. Why do you need so many sperms? Because not all of them will be absolutely normal in shape, size and structure. May not be. At the same time, they should have a particular amount of motility for them to reach till the fallopian tube where the ovum is present, because you know that the ovum is not viable for more than 24 hours. So fertilization has to take place in that crucial time period. For that, there are 200 to 300 million sperms, and for efficient fertilization, important MCQ which you need to remember is that for efficient fertilization, we need 60% of the sperms to have normal shape and size, and of these, please pay careful attention, of these 60%, not of the 200 to 300 million, of the 200 to 300 million, 60% should have normal shape and size, and of these, 40% should show vigorous motility. So practically 24% of the sperms should be absolutely normal in shape, size, structure and have vigorous motility so that fertilization efficiently takes place. Is that absolutely crystal clear? Cool. So moving ahead, now once sperms are formed, this can come as your question. It is a PYQ that had come, my dear children, where they had asked what is the correct order or the correct sequence of transport of sperms through the male reproductive system. So once they are formed in the seminiferous tubule, they will go to the retitestes, from there to vasa efferentia, to the epididymis, to vas deferens, ejaculatory duct, urogenital duct, that is urethra, and then through the urethral meatus outside. So this has come as a PYQ. 2018 question paper. 2017 you had the same question but with respect to frog. 2018 you had the same question with respect to human males. Okay. So let's see the question. Select the correct sequence for transport of sperms in the male reproductive system. Yes, so can I have answers quickly? What would be the correct order? The correct order would be, is it testes to epididymis to vasa efferentia retitestes? No, no, no. Seminiferous tubules to retitestes to vasa efferentia to epididymis to vas deferens, ejaculatory duct, urethra and urethral meatus? Yes, this is our correct answer. Yes, very good everyone. So the correct answer is option key B. Okay, very good. Moving ahead, if there are 100 million sperms in an ejaculate, how many sperms are essential for efficient fertilization? Now, we just saw that there has to be 60% of the sperms which have normal shape and size, and of these, 40% should show vigorous motility. So that means it has to be 24% of the sperms. So if this was 200 to 300 million sperms, we needed to calculate. However, if they are given 100 million sperms, the answer is quite kind of, very simple and direct, where you have 24 million sperms which are having normal shape, size, structure and vigorous motility for efficient fertilization. So that was about male reproductive system. Moving ahead to understanding of female reproductive system. Now this is again the sectional view of the NCERT itself of the female reproductive system, where you can see from ventral to dorsal you have pubic symphysis, followed by the urinary bladder, followed by uterus, where you can see fallopian tube, and the ovaries, that is the primary reproductive organ, and dorsal to it is the rectum, and here is the anus, followed by is the sacrum. So again, if we talk about the female reproductive system, it is situated in the pelvic region, where you have primary reproductive organs, that are ovaries, followed by accessory reproductive organs where we have a pair of fallopian tubes. We have uterus, including uterus, the lower part of the uterus is the cervix, and the external the copulatory organ, which is vagina, followed by the external genitalia. Now, like in males, we saw that testes' main function is production of male gametes, that is sperms, and androgens, that is the sex hormone. Similarly, in females, again, ovaries, which are primary reproductive organs, which are situated in the lower part of the abdominal system. Again, here, their main function is production of the female gametes, that is ova, and at the same time producing female sex hormones, which are group of two hormones, which is estrogen and progesterone. Again, like androgens, these are group of hormones. Keep remembering these small points. Now, how do these produce gametes? Now, when we are talking about female reproductive system, there is a slight difference with respect to male reproductive system. In male reproductive system, we saw that at puberty was the process that started. Now, that process starts at puberty, continues till the last day of the male life. But if we are talking about female reproductive system, the process of oogenesis, that is formation of ova, starts in the fetal life itself and will only be completed once fertilization takes place. So, in the fetal life, there are these egg mother cells which are called as oogonia. Now, these oogonia are diploid cells. These oogonia will now undergo mitosis followed by differentiation to form primary oocytes. Now, since these primary oocytes are formed by mitosis, these are also diploid cells. They will undergo their first meiotic division, which is reductional division. Now, remember, in the fetal life, they start their meiotic division, but it is not completed in the fetal life. So, the cells are still called as primary oocytes, which are present in the form of a couple of million of them at birth. Till puberty, some of them degenerate for reasons not known, and at puberty we have approximately 60,000 to 80,000 primary oocytes present in each ovary. Remember, it is in each ovary. So, these will then go into the form of follicles. So, these primary oocytes initially in the form of primordial follicle. However, the word primordial is not given in the NCERT. Then at birth you have primary follicles which are present, wherein there is this primary oocyte surrounded by a layer of granulosa cells. At puberty, what happens is there is FSH that is released. This FSH, follicle stimulating hormone, I'm extremely sorry. This follicle stimulating hormone will act on the primary follicles, converting them into growing primary follicle, into secondary follicle, and where secondary follicle will develop a layer of cells around it called theca cells. Then the secondary follicle will convert into tertiary follicle, where you have an outer layer of cells which is called theca externa and an inner layer of cells called theca interna. It is this theca interna that starts to now secrete a group of hormones called estrogen, which now also develops a small cavity here, which is a characteristic feature of tertiary follicle called antrum. And the primary oocyte, which is present in this tertiary follicle, still halted at the diplotin stage of prophase 1, is now pushed little eccentric in position. Further FSH comes, converting the tertiary follicle into the most mature follicle called Graafian follicle. Again, we have on the outer side theca externa, inner side theca interna, which is secretory in nature. The antrum increases in size, and just before the tertiary is converted into Graafian, remember, just before the tertiary is converted into Graafian follicle, the primary oocyte which was there, halted at diplotin stage, now quickly completes its meiotic division, that is the first meiotic division. But there is a difference. The first meiotic division here is unequal, where there is a large haploid secondary oocyte and there is a small first polar body that is formed. Why is there a difference in the cytoplasmic content? The nuclear content is equally divided. 23 chromosomes in the first polar body, 23 chromosomes in the secondary oocyte. So then why the cytoplasmic inequality is because here basically this secondary oocyte, which will now get converted eventually after meiosis 2 into the ovum, which we'll see in some time now, retains the major bulk of cytoplasm, because at the time of fertilization, now this is a question in the NCERT, so there is a direct answer that I'm giving you. The secondary oocyte which is there and the ovum retains the bulk of the cytoplasm, because during fertilization it is only the nucleus and the proximal central from the sperm that enters into the ovum. Okay, so all the other contents which are there of the cytoplasm needed for further cell division are all present in the ovum, and hence the cytoplasm of the ovum and the secondary oocyte is major or is retained in large proportion as compared to the first polar body, and it's the first polar body whose function exactly and the second polar body is not known till date as per given in the NCERT. Now this secondary oocyte, which is the first haploid cell in the entire process, will start its meiosis 2 again, but it will again halt. Halt at what? Halt at metaphase 2. Till when? Now if you read fertilization part from the NCERT, it is halted till the time the first sperm comes and touches the zona pellucida. Okay, so the first sperm when tries to fertilize the ovum, that is the time the secondary oocyte, which is halted at metaphase 2, will quickly complete its meiosis 2, again an unequal division, forming a large haploid ovum and a small haploid second polar body. Is that absolutely crystal clear? So, unlike spermatogenesis, which started at puberty, continues till death, here it starts at the fetal life, halts, and is completed only when fertilization takes place. So now what happens if the fertilization does not take place? Let us see and understand it with the help of menstrual cycle. Now what is this menstrual cycle? It is basically a cyclical process in every female primate, and female primate includes monkeys, apes and humans. This reproductive cycle in female primates is called as menstrual cycle. It starts with a one menstrual phase or menstruation where there is bleeding that takes place to the next menstruation, which lasts for approximately 28 to 29 days, and hence a regular cycle is a 28-29 day cycle. It's cyclical events that occurs, as I told you, from one menstruation till next menstruation, called menstrual cycle. Now when does it start? Again it starts at puberty, and that phase is called as menarche. However, it will not continue till the last day but stops at the age of around 50 years, and this is called as menopause. So the time period from menarche to menopause is basically the only time where the female is capable of reproduction. So let us see through this graph and understand it beautifully. There is a graph given in the NCERT. Yeah, so now we have this entire menstrual cycle divided into phases. What are the phases? You can see there are a number of days which are mentioned here. Here you have the lowermost section which talks about the endometrial thickness, followed by the ovarian hormones, changes in the ovaries. So this part is entirely what is happening in the ovaries with respect to the follicles, with respect to the hormones, and here we have the pituitary hormones. Okay, so if you see from day, so this is basically the first phase, which is 3 to 5 days. Then on the 14th day is when the ovulation takes place. We'll talk about this, and then you have the phase. So the first phase, which is the first 3 to 5 days phase, is the menstrual phase, followed by the follicular phase or the proliferative phase. Then the shortest phase, which is on the 14th day, is the ovulatory phase, followed by the secretory phase. So let us see. Now if you're talking about menstrual phase, this basically happens when the female has not conceived. When there is no fertilization that has taken place, the endometrium sheds off. Why did the endometrium thicken? We'll talk about it in some time. The endometrium sheds off. You can see there is bleeding that is shown here. That is, there is menses that is taking place. Once the bleeding phase takes place, it lasts for 3 to 5 days. followed by which is the proliferative of the follicular phase. Now during this phase, FSH and LH hormones slowly gradually increase, which bring about maturation of the follicles, primary to secondary to tertiary and eventually Graafian follicle. Now once the Graafian follicle is formed, you can see that from the tertiary follicle and the Graafian follicle, the theca interna has started to release estrogen. So there is small amount of estrogen level that keeps on increasing. On the 14th day, there is peak of estrogen that occurs. There is also FSH that has reached its peak, and there is a sudden surge in the amount of LH. This sudden surge or increase in the amount of LH cannot be borne by the Graafian follicle and it ruptures. This phase, when it ruptures, leads to release of ovum in the stage of secondary oocyte process called ovulation, which lasts for approximately only 1 day. That is the shortest phase of the menstrual cycle. Once the rupturing takes place, once the ovulation takes place, the theca interna cells are now ruptured, the Graafian follicle is ruptured, and the estrogen level comes to its baseline. Similarly, FSH also comes to its baseline. LH, which had gone into a surge, reduces but does not reach its baseline. See, that is because this LH helps in maintaining a structure called corpus luteum. The ruptured Graafian follicle within 24 hours will now get converted into a yellow colored mass or body called corpus luteum. It is this corpus luteum that slowly gradually starts to release progesterone. As progesterone is released, it slowly gradually thickens the endometrium. Why? It is preparing the endometrium for the purpose of implantation if fertilization has to take place. But the later phases of the secretory phase, or the luteal phase where the corpus luteum is formed, there is another peak of estrogen that takes place, and estrogen aids the progesterone. Progesterone is the main hormone to maintain the thickening of the endometrium or the thickness of the endometrium, and estrogen just helps to maintain that thickness. Now this will be maintained for a period of 14 days. If in the 14 days fertilization takes place, fertilization has to eventually take place on the plus minus 24 hours. That is, once ovulation takes place, within 24 hours the fertilization has to take place. If fertilization has not taken place, still the corpus luteum will be maintained for 14 days, post which it will shrink to form a white colored mass called corpus albicans. Now this term is not given in the NCERT, if you can remember, well and good. So corpus luteum, once it degenerates and shrinks, there is nothing to secrete estrogen and progesterone level drops. As progesterone level drops, the endometrium sheds off, and that marks the beginning of the next menstrual cycle. Is that clear? So there is a cyclical menstruation that continues from menarche to menopause every 28-29 days, and this is an indicator of the normal reproductive phase. Now, when we talk about reproduction, reproduction basically means that there is production of new organisms of the same kind or the same species. Now, there is another topic that we go into or dwell into, which is reproductive health. Now, with respect to reproduction, when we are talking, maintaining the health of the reproductive system is utmost important. Why? Not only at the personal level, but if the person is healthy reproductively, then it will lead to a reproductively healthy society. So NCERT says that WHO gave us a definition saying that reproductive health is a state of complete physical, behavioral, social and emotional well-being in all aspects of reproduction. Now, one of the major problems that India was facing with respect to reproduction was that there was an extensive population explosion post-independence. So, in the year 1951, Government of India launched a nationwide program which was called Family Planning Program, whose initially the main focus was reproductive health and at the same time curbing and controlling the population explosion. Because if you see the population rate, statistically the growth rate is extensive. If you are talking about the world population, 1900 the world population was 2 billion. 2000 it increased to 6 billion and 2011 it rocketed to 7.2 billion. India was nowhere behind. At the time of independence, it was 350 million just. However, 2000 1 billion, and now 2011 1.2 billion. This year, 2000, every 10 years statistical data is done, census is done, we wait for the census now. But however, if you see 2011, it is 1.2 billion, which is a huge expansion. So government eventually, along with maintaining the reproductive health, revamped and revised its program, and now called as Reproductive and Child Healthcare Program, which mainly focuses on controlling the population explosion. One of the methods that it does is with the help of contraceptive methods. Contraceptive conception, so prevents conception. Now, what is an ideal contraceptive method or an ideal contraceptive? An ideal contraceptive is the one who is user-friendly, easily available, effective, at the same time reversible, least or no side effects, and in no way interferes with the sexual desire or the sexual drive of the person. We broadly classify the contraceptives in two methods. One is temporary contraceptives and one is permanent contraceptives. Temporary contraceptives, we include natural or traditional methods, IUDs, oral contraceptives, injectables and implants. And in permanent, it's a surgical method. Let us see one after the other. When we are talking about natural method or traditional method, here there is no use of any external means or medications. Hence, the side effects are very less. So how does it work? It basically works by preventing the meeting of the gametes. So if you are not allowing the gametes to meet, obviously fertilization does not take place and conception does not take place. One of the method is periodic abstinence. Now we just studied menstrual cycle, where we studied that in a 28-29 day cycle, the ovulation usually takes place on the 14th day. However, it is not by the calendar that every 28th day, that is every 14th day of the menstrual cycle, the female will ovulate. So it can be a plus minus a few days. So if you are saying 10 to 17 days is the ideal time period where the female will ovulate. Now in this time period, the chances of fertilization are very high. So you advise the couple to abstain or to prevent from having coitus during day 10 to day 17 of menstrual cycle, which is the day which is also called as the most fertile period. Previous to this and post this, however, previous to this, the ovulation has not taken place. Post this, the ovum is dead. So there are no chances of fertilization. Second method is withdrawal method. Now in this method, the male partner is advised to withdraw the penis from the vagina just before ejaculation takes place. So if the male is withdrawing the penis, there is no ejaculation of semen. If there is no seminal ejaculation, sperms do not enter the female reproductive tract and fertilization does not take place. The next is lactational amenorrhea. In lactational amenorrhea, lactational at the time of when the female is lactating. Amenorrhea, absence of menses. Now this is the time period where there is no menstruation taking place when the female is lactating, that is breastfeeding the baby. This is because when the female is breastfeeding the baby completely, that is the time there is an inhibitory effect on FSH and LH, thereby follicular maturation does not take place, ovulation does not take place, and if ovulation does not take place, there is no question of fertilization. So that means that is the entire period when the female is lactating, she will not be able to conceive? No. It is only till the time that the female is completely breastfeeding the baby. That is a period of maximum 6 months following parturition, and this has also come as a PYQ. Moving ahead to the next method, where we talk about barrier methods, condoms, cervical caps, diaphragms and valves. When we are talking about condoms, condoms are available, these are basically made up of thin rubber latex material. They can be used by males as well as by females, and their main purpose is to cover the penis in males, cervix or vagina in females, thereby physically preventing the gametes from meeting. Then there's cervical caps, diaphragms and valves. These are used only by females. They cover the cervix just before coitus it is used, so that the semen will not enter into the female reproductive tract and there will be no fertilization. Then, to increase the efficacy of cervical caps, diaphragms, walls or barrier methods, we can also advise a couple to use spermicidal creams, jellies and foams which are spermicidal, that is they will kill the sperms. Moving ahead to the next method, which is IUD. IUD is intrauterine devices which are inserted within the uterus. For this, there is need of an experience or an expertise of the doctor or the medical staff, that is the nurse, which is required to introduce this into the female reproductive tract. It is one of the widely accepted method to space children or to delay pregnancy, and this was again a PYQ. This is the part from where a large number of questions are coming. IUDs are of three types. We have non-medicated IUDs, copper-releasing IUDs and hormonal IUDs. Example of non-medicated. Now, all of these IUDs will increase phagocytosis. However, hormonal ones and copper-releasing ones have an additional function to do. So, example of non-medicated is Lippes loop. Copper-containing is copper T, copper 7, multi-load 375, and hormonal is progestasert or LNG20. Now, non-medicated will just bring about phagocytosis of the sperm. However, copper-containing will keep on releasing copper. As they keep on slowly, gradually releasing copper, copper will reduce the sperm motility and the fertilizing capacity of the sperm. Thereby, it will prevent the sperms from easily reaching the fallopian tube and thereby prevent fertilization. However, hormonal will make the cervix hostile to the sperms and the uterine endometrium unsuitable for implantation, along with increasing the phagocytosis of the sperms. Then comes OC pills. Oral contraceptive pills. Contraceptive pills which are taken orally, hence called oral contraceptive pills. Okay. The commonest one used is Saheli. Now, these oral contraceptive pills obviously are used only by females. How do they work? What do they contain? So, they basically contain either only progestogen or a combination of estrogen and progestogen. Now, these are pills which are taken by the female for a period of 21 days, starting from the first 5 days of the menstrual cycle. So, once she starts menstruating, within the first 5 days, she will start taking the pills. Take it for a period of 21 days. Post that for 7 days, she stops the pills, and it is this time that she menstruates. Meaning, it is these pills which are inhibiting ovulation, but they will make the endometrium thicken. So, during the 7 days when she is not taking the pills, the endometrium sheds and she will bleed. So, during that 7 days of gap, she will have her menses, and when she has started with her menses, the first 5 days, she starts taking the pills again and continue it for the next 21 days, stop for 7 days, again start in the first 5 days of menstrual cycle, continue it for 21 days. For how long? Till the time that she wants to prevent her conception and thereby act as a contraceptive method. So, this basically inhibits ovulation, as I told you. At the same time, it also makes the cervical mucus hostile and alters the quality of it so that it can prevent or retard the entry of sperms. They are very, very effective. They have lesser side effects and well accepted by females. However, few side effects are there. So, to prevent that, the government came up with another contraceptive pill which is called Saheli. This was discovered by scientists at CDRI Lucknow, that is Central Drug Research Institute Lucknow. This can come as our MCQ. It is a non-steroidal pill. Does not contain any steroids, that is, does not contain any hormones. It is non-hormonal. It need not be taken every day. It has to be taken only once a week. It has very few side effects and high contraceptive value. So, this is again the one which is commonly used. The same hormones, that is either progestogen only or estrogen progestogen combination, can be used either in the form of injections or in the form of implants by the female. These injections, implants, or IUDs, when used within 72 hours of the unprotected sexual intercourse will act as an emergency contraceptive. Then comes a permanent method, which is vasectomy and tubectomy. Now, when you're talking about vasectomy and tubectomy, these are permanent methods wherein a small surgical procedure is required. So, it's a surgical method. And it's permanent. Their reversibility is extremely poor. In males, we call it vasectomy. In females, we call it as tubectomy. Ectomy means surgical removal. So, there's a small cut that is made on the vas deferens. So, the small incision means a cut is made on the scrotum and the vas deferens is ligated and cut. So, what are you doing is you are preventing or blocking the transport of gametes. So, gametes from the testes will now not enter in further into the male reproductive system post vas deferens, and the semen will be without sperms. So, even if ejaculation takes place, there are no sperms in the semen, it's only seminal plasma. So, there will be no chances of fertilization. Similarly, in female, a small incision is made on the lower abdomen or via the vagina. A small part of fallopian tube is cut and ligated, and thereby the ova which is there will not move, nor will sperms reach towards the ovum. So, you are blocking the transport of gametes. However, once this is done, it's a permanent method where the couple now does not want to have children, and this can be done either of the partners because then the reversibility is very, very poor. What are the side effects? The side effects are nausea, abdominal pain, breakthrough bleeding, irregular menstrual cycle, breast cancer. So, periodic abstinence is one such method in which the couple avoids or abstains from coitus from day dash to dash of menstrual cycle. The answer is day 10 to day 17, which is the most fertile period. Moving ahead to the next question. Assertion and reason. Sterilization is a terminal method that can be used by both males and females, and reason is sterilization is highly effective method and its reversibility is very, very good. So, let us see what is the answer. So, assertion is sterilization is terminal method, that is permanent method that can be used both by males and females. Yes, it's a true statement. However, sterilization is highly effective method and its reversibility is good. No, it is vice versa. Reversibility is very, very poor. So, assertion is true and reason is false. So, that is from my side about reproductive health. Moving ahead to understanding of health in general. Now, there are large number of pathogens which are surrounding us. Do all pathogens cause disease to us? Yeah. So, when we are talking about immunity, do we suffer from all these diseases every time? No, we are not going to suffer from disease every time because God has provided our body with a beautiful defense system, and that is called as immunity. Now, what is this immunity? This is directly from the NCERT, so that I wanted to highlight what is given in the NCERT. An overall ability of the body to fight against the disease-causing agent or organisms conferred by the immune system is called as immunity. Now, this immunity which God has given us is of two types: acquired immunity and innate immunity. Now, let us talk about it. Innate means inborn. This is also called as inborn immunity. And acquired immunity, as the name suggests, is acquired during the lifetime of an individual. This is the one that we are born with. This is the one that we gain during the lifetime of an individual. Now, innate immunity is non-specific immunity because it provides protection against any kind of infection. How? We'll talk about it in some time. Whereas, if you're talking about acquired immunity, it's highly specific because we will see that it is with the help of antibodies that are produced by the body. These antibodies that are produced, they are specific for specific pathogen, and the same antibody will not work against every pathogen. Hence, it is highly specific. Comparatively, the innate immunity is faster to act, and the acquired immunity which is there is comparatively slower. It works via barriers, that is physical, physiological, cellular and cytokine barriers, and acquired immunity is via clones of T lymphocytes and antibodies. Now, what are these physical, physiological, cellular and cytokine barriers? When we are talking about physical barrier, barrier means protection, it is preventing, it is blocking, okay. Physically preventing the pathogen from entering the body, which is via skin, which is via the mucous membrane which is present in the respiratory, the digestive, the urinary and the genital, that is the urogenital system. So it traps the foreign particle and prevents it from entering inside. Then comes the physiological barrier. Physiology means normal functioning. Physiological barriers are the ones such as acid in the stomach, the pH of saliva. There are lysozymes in the tears. These are antibacterial agents, the lysozyme. Acid will prevent the pathogen from further entering deep inside and thereby act as a barrier. Then comes cellular barrier. There are certain cells. Which are the cells? We have WBCs, but not all WBCs will fall into this category. We have PMNL, polymorph nuclear leukocytes, which are neutrophils, monocytes, and natural killer cells type of lymphocytes in the blood, as well as the macrophages in the tissues that can phagocytose and destroy the microbes. Then comes cytokine barriers. These are basically proteins which are secreted by cells which are already infected by viruses. So they do not want their neighboring friends and cells to get infected by virus. So they will secrete these chemicals, these proteins called cytokines, which protect the surrounding non-infected cells from suffering from these viral infections. Yeah. So let us quickly take a question on this. So in column 1, we have physical barrier, physiological barrier, cellular and cytokine barriers, and on the other side is the examples. So physical barrier, what will be the answer? Physical barrier obviously will be our skin. Physiological is fever, cellular is PMNL, polymorph nuclear leukocytes, cytokine barriers is interferons. So answer should be 1, 2, 3 and 4. Now when we are talking about acquired immunity, that was about the innate immunity. When we are talking about acquired immunity, we have two types of responses called primary response and secondary response. Primary response, when the body encounters the pathogen for the very first time, the response that it produces is called primary response. Similarly, when the body encounters the pathogen which is the exact same pathogen consequent or subsequent time, we call this kind of immunity, this kind of response as secondary response, or it is also known as anamnestic response. What do we call it as? Anamnestic response. Yeah. Now this anamnestic response which is there is comparatively faster and is of a higher intensity, hence anamnestic. Whereas this is of comparatively slightly lower intensity. Now what are the cells that provide immunity? We have two types of cells called B cells, that is B lymphocytes. The cells are basically the lymphocytes. So we have B lymphocytes and we have T lymphocytes. B cells commonly called and T cells. These are lymphocytes. Now B cells which are there are the ones that produce antibodies, and T cells are the ones that basically form clones, which are 4 types, which are helper T cells, killer T cells, suppressor T cells and memory T cells. So it is via the cells that they will provide immunity. These are via antibodies. So since they are providing immunity, B cells via antibodies, you call them as humoral immunity, or you call it as antibody mediated immunity. Whereas the immunity provided by T cells is called as cell mediated immunity, or we call it as cellular immunity. Now let us talk about the structure of antibody. It's a Y-shaped structure which is made up of 4 polypeptide chains, where there are 2 heavy chains and 2 light chains called as H2L2, 2 heavy and 2 light chains. These are joined together, the heavy to the light, the heavy to the heavy, and the heavy to the light are joined together by disulfide bond. Each antibody has a part which is called as antigen binding site, the one that you can see here. And it is this place where the antigen comes and binds, forming an antigen antibody complex, which is a lock and key mechanism, and thereby producing further immunity. Now when we are talking about types of acquired immunity, there are 2 types. There is an active immunity and there is a passive immunity. Now what do we mean by the word active? Active means the one that is performing work. When the pathogen enters into the body and body produces its own antibodies, you call it as an active immunity. When the body receives ready-made antibodies, you call it as passive immunity. Both of them, we have 2 categories. In active, we have naturally acquired active immunity. We have artificially acquired active immunity. In passive, we have naturally acquired passive immunity. We have artificially acquired passive immunity. When we are talking about naturally acquired active immunity, this is the time when we have any kind of infection into the body and the body produces its own antibodies. Then it is naturally producing antibodies because of a natural stimulus, so naturally acquired active immunity. When we provide or give any kind of vaccine to the person, except 3 that we'll be talking about here, we are talking about artificially acquired active immunity. Then there's naturally acquired passive immunity. When we are talking about naturally acquired passive, means naturally body is receiving antibodies. When will this happen? Say for example, from the mother to the child, it is only mother who gives you everything for free, isn't it? So from the mother to the child, either via placenta or from the mother's milk, that during lactation. So that is from the mother to the child, either transplacental or it is via lactation. And when we are talking about artificially acquired passive immunity, means in such diseases where you cannot further give vaccines, you will have to give them a curative vaccine. In such cases, say for example, you are talking about tetanus or rabies or anti-venom in cases of snake venom. Body is receiving, we are giving ready-made antibodies to the person, then you call it as artificially acquired passive immunity. Is that clear? So quickly let us take the question. Yes, very good, Mayank. Antigen, killed or attenuated, introduced during a vaccine, stimulates cell mediated. So you're giving an antigen into the body through a vaccine, I just explained you, produces cell mediated as well as antibody mediated immunity and memory cells. This is an example of naturally acquired active, naturally acquired passive, artificially acquired active or artificially acquired passive. Now you are giving a vaccine, means is it naturally happening or you are deliberately doing it? You are deliberately doing it. What are you giving? Are you giving antibodies or you are introducing antigen? You are introducing antigen. So it is artificial, 1. Is the body producing antibodies? Yes. So it's artificially acquired active immunity. Is that clear? Very good. Moving on to the last part of the topic where we are talking about drugs. Before that, just give me a second. I'll have a sip of water and then we proceed. Okay. So when we are talking about drugs, so these drugs, as we say, or we call them as drugs, they're basically the ones which are either used medicinally or for any kind of religious purpose since ages. But when these are used for purposes which are not medicinal or in doses which are not medicinal, you call it as drug abuse. The commonly abused drugs are obtained from plant sources, except one which is from an ergot fungus, which is Claviceps purpura. That's mentioned in the NCERT, not the name of the drug, but it said that most of them are plant sources except this one which is obtained from fungus. The commonly abused drugs are opioids, cannabinoids and coca alkaloids. For making it very easy for you to study, since students find it little difficult and tricky and confusing, I have compiled it in the form of a table which I'm sure is going to make it very easy for you all. Now, we are going to talk about with respect of the drugs with respect to it, where it is obtained from, what is it commonly called, if there is a name to it, where does it act into the body and what is its effect. Because this is the part, lot of questions come from here, okay. So, if you're talking about opioids, okay, this is the structure of the drug obtained from opioid. What do we use, or which part do we use? So basically it is obtained from latex of the poppy seeds, okay, of the poppy plant, which is Papaver somniferum. And what do we obtain is a drug which is called as morphine. Now this morphine, when we chemically treat it and undergo an acetylation, it becomes diacetyl morphine. And this diacetyl morphine is called as heroin. It's called as heroin. This is the structure of opioid drugs which can come as our MCQ. What is it commonly called? It's commonly called as smack. Okay. Where does it act? It acts on the central nervous system obviously and also on the digestive system. It slows down the functioning of the nervous system and the digestive system, and it acts as a depressant. It's commonly used medicinally as an excellent sedative and a painkiller post major surgeries, especially abdominal surgery. So once a person has undergone abdominal surgery, you can understand there would be an excruciating amount of pain and suffering. So to reduce that, to calm the body, to kind of let the person rest and relax, you give them morphine. However, when it is abused, then you call it as drug abuse. Okay. Then comes cannabinoids. Okay, now cannabinoids are obtained from a beautiful plant which is called as cannabis sativa. It is the inflorescence of this plant. You understand the meaning of the word inflorescence? Botany, you all are experts in botany also. So inflorescence of cannabis sativa plant, and what you obtain is cannabis. But there are a lot of other combinations also which you can obtain from this plant, such as hashish, charas, ganja, and also marijuana. These are obtained from flower tops, leaves, and also from resin. There is no as such common name given to it. They are commonly called by these terms only. It acts on the brain, that is the nervous system, and the cardiovascular system. Okay, and it basically kind of activates these systems. Then we are talking about coca alkaloids. Coca alkaloids is obtained from Erythroxylum coca plant. This plant is native to South America. Common name is coke or crack. Okay. So it interferes with the transport of neurotransmitter dopamine, and this can come as our MCQ. So this is important. Okay. It acts as a stimulant, and in excessive doses, it also acts as a hallucinogen. There are other hallucinogens such as atropa, belladonna and datura, along with coca alkaloids or cocaine, which are also acting as a potent hallucinogen. Okay. So that is about a summarization and a beautiful presentation of all the drugs. You need to remember all of this. Simultaneously, you also need to remember the structure of opioids and structure of cannabinoids. Okay. Let us quickly take a question on this. Identify the molecule A and molecule B and select the correct answer. Let me see how many of you all answer. Yes. Okay. We've just seen the structure some time back, almost few seconds ago. Showing it to you all again. This is opioid, this is cannabinoid. So what would be this? These are opioids. This is cannabinoid. So A is cocaine? No. B is heroin? No. B is cannabinoid? Yes. So let us see B cannabinoid. Source is Atropa belladonna. No, it is cannabis plant. Produces hallucinations, higher doses. A is morphine. Yes, we know morphine and heroin. So A is morphine. Yes. Source is papaver somniferum. Yes, it's a very good sedative and a painkiller. So answer is D. So that's all from my side for today. So I hope you've understood spermatogenesis, oogenesis and menstrual cycle absolutely thoroughly well. You've understood the contraceptive methods from which every year almost without fail one question is coming. And you've also understood the immunity part, okay, and the drugs, which was quite beautifully summarized for you all. So I hope today's session was absolutely crystal clear, was very informative, very helpful for you for the NEET exams. So with that saying, all the best for your NEET preparations. Do well, rock it, and I hope all of our sessions are very, very helpful for you all for the upcoming NEET exam. So take care, study well, see you soon in the next session. Thank you very much and see you for today.